break, please, those who will be presenting later and have not delivered yet their presentations, please do that during that period of time to avoid delays and to waste time. Then we return here. We'll see whether there are still some issues from the lectures that are required to be revisited. And if not, I will run through the papers for the next day, okay? And then we will make the assignments and all that. And then after that, we will start with the group presentations, okay? So as we decided the other day, and not all of you were here, but the idea was to have 4 or 5 teams, and then each team will receive each day 1 paper, and then next day, someone of the team has to make a presentation of that paper. The last week we had a wonderful aperitif by 2 of your colleagues, and then I hope that we can maintain the level and the quality of the different presentations of the kind of journal club that we will have the last hour of our sessions per day. Okay, so I want to spend 2 days, today and tomorrow, and maybe a bit on Wednesday, to discuss 3 very central concepts in synthetic biology that, you know, if at the end of this course you just remember what is 3 words that I'm going to discuss with you today, tomorrow, and the day after tomorrow, I will be more than happy. Okay, so but please just keep in mind that the today, tomorrow, and the day after tomorrow probably will be the most important classes of the whole course. Okay, so we start today to address the problem or the issue of the jargon. So jargon means what you see there. Jargon means a specialized language that is concerned with a particular subject, and it contains also a specific term and so on, but also jargon is a word that is usually said to describe a language that is characterized by pretentious syntax and also by gibberish. So when they say jargon, jargon is the collection, the specific terminology that we use in synthetic biology. But just keep in mind that sometimes this terminology is abused and people use it in ways that should not. Okay? So the first super central concept of this jargon in synthetic biology is the concept of orthogonality. Orthogonality is a mathematical concept and means what you have there, namely that 2 systems are orthogonal to each other if they do not influence each other. This is a mathematical concept that has been brought into synthetic biology in a kind of analogy, and you will see later how this term fits in the whole concept of engineering biology that is at the core of synthetic biology. But let's start from the beginning. So in the last 20 years, the big change in biology has been the change from genomics, from genetics to genomics. Genetics means looking at genes one at a time, one after the other, making a mutant, see what are the consequences of the mutation. Genomics is about looking at all genes simultaneously, okay? And this is something that has been enabled by various technologies that do not belong to the course that we are running, but you are aware of them, like proteomics, metabolomics, all types of omics. But the core, scientific discipline that is connected to all these technologies is genomics. Genomics means doing things at large scale in many respects. Okay, I'll show you just a few examples of how this works, how you can move from genetics to genomics. So for instance, in the times of genetics, and well, it is not that we don't have genetics today, but the traditional genetics would, for instance, identify the function of a gene on the basis of having a mutant and then examining the phenotype. Now, under genomics, what you do in many cases is just look at the sequence. You don't make experiments, you don't make knockouts in many cases. You just look at the sequences, and by looking at the sequences, you in some cases can predict with a high degree of confidence what is the function of that particular DNA sequence. And I would like to remove that, but we have always the same problem. Right? Let me see just 1 second. Well, I'm sorry, but I don't know how to get rid of the upper panel, so we may have to live with it. Well, in any case, so what I was saying is that in genomics, in many cases, the identity and the function of the genes can be derived, can be deduced from the DNA sequence, okay? Now what? Better to be at this. Okay, so also what is says here is the number of regulators that you find in different biological systems, in different bacteria in this case. So it is quite evident for these studies where you can predict the number of genes and the number of regulators that you have in a biological system or in a bacterium just by looking at the sequence, that not all bacteria have the same number of regulators. So some bacteria, like Mycoplasma for instance, have basically no transcriptional regulators, but there are other, say, bacteria, like Pseudomonas aeruginosa or E. coli that have a very large number of regulators. So that means that we are really visualizing through this means a very large or a very high level of complexity. Also, the number of regulators that you have in a bacterium runs exponentially in respect to the number of genes. Okay? So in most cases, the larger the genome is, the more genes we have, right? But the number of transcriptional regulators, instead of following a linear relation, what it follows is an exponential relation. So that means that the bigger the biological system is, or the more complex genomically, the more transcriptional regulators we have. Now, how are these transcriptional regulators organized? Well, they are organized in many cases, not in isolation, but forming regulatory nodes, as you see here. So in a regulatory node, typically you have inputs and outputs. Remember what we discussed a bit in the previous classes. So in the simplest configuration, you have a single input and you have a single output. But in other cases, you may have a single input and various outputs. And this is typical when you have a transcriptional regulator that activates or represses various target genes. But at the same time, you may have devices, genetic devices, existing naturally, where you have various inputs that are computed to produce a single output here, and you have the final combination where you have various inputs that deliver various outputs. So you have all types of possibilities that, similarly to what happens in electronic circuits, can be observed also in biological systems. So the way this has been represented typically has been with these arrows. You have different ways of connecting, of abstracting the different types of regulatory motifs, regulatory networks, and then, well, you can, this is the typical, say, textbook depiction of how different regulators will interact with different target genes. And what happens is that depending on the type of regulators, then they can establish simple, say, connections between them, or they can be really, really complex. And we will return to that at a later point. Now, as we discussed last week, the essence of synthetic biology is looking at biological systems as if they were engineered systems and following the same logic as engineered systems. So if that is the case, we should find similarities between complex regulatory networks that we observe in bacteria or in biological items and regulatory networks that you find in electronic devices. So for instance, in this case, this is an information processing scheme, different layers of Linux, the code graph. You have a master regulators are here, the middle managers are here, and the workers are here. And it is interesting to make a comparison between this type of layering in the regulation of a man-made, say, device and what you observe in the bacterial world. So this is what you have in bacteria. You have a master regulator that will be equivalent to this master regulator that you observe in electronic circuits as well. And you have this middle manager, and you have this workhorse. So as you see, the difference here is that you have many master regulators, you have many middle managers, and you have just a few workhorses. In the case of biological system, you have just the contrary. You have a limited number of master regulators. In fact, it's only about 10 middle managers. You have some of them, and then at the end, you have all these workhorses that are actually the genes that are being activated or not by the regulatory system. So what type of typical, say, motifs you find in these transitional regulation networks? Well, the most, say, let's call it famous type of network motif is the so-called the feed-forward loop. So these feed-forward loops were identified by someone called Ri Alon, like 15 years ago, and they go as follows. So you have like three elements in the biological system, X, Y, Z, and they can interact with each other. They can say interplay with each other in many different positive or negative conditions. So you have in that way you have two types of feed-forward loops. They can be coherent or incoherent, and well, you have a list here. I will not go into detail one by one. But basically, the most typical regulatory node that you find in nearly every regulatory, say, element is a situation where you have one element acting positively or negative on a second one, and then either having a third element acting positively or acting negatively. And you can make all types of combinations. Okay? And each of these situations can be modeled and represented mathematically, and they predict behaviors that can be observed very easily in natural regulatory networks. Now here comes the treat for synthetic biology and for biotechnology in general. Okay? You have a pre-existing regulatory network with genes, with tips, with all types of. So how do you manage to enter into this very complex regulatory network a new actor? Okay, so you may think that all you have to do is to from the gene put it into a cell, but then the cells, for the most part, may not like that. And if you do just that, in most cases, it will not work. Okay? So the question here is how do you manage a situation where a new component of a regulatory network that carries along a gene of interest will be able to, say, nest and to remain there for a short period of time for delivering a given function that we want to do. Okay, so we can take a little abstraction and say, well, the situation that we start with is this one. So you have all these complex situations where you have various partners, and you see all partners are connected to the others because they share some surfaces. These surfaces can be physical or can be just functional. Okay? So you have a geometrical form that represents activity coupling or say activity balance, and well, this is what you have at the moment. However, what happens if you want to replace or add one more member of this complex to this preset assembly, given that this new member doesn't really fit? So it's like you want to fit a triangle here, and well, how do you fit a triangle here? There's no way you can fit a here, because the architecture of the place is such that you need a quadrangle, you don't need a triangle. If you want to put a triangle there, it will work. And if it works, it will work only for a short period of time because it will be sooner than later kicked off from. So there are two possibilities to handle this situation. Possibility number one is to make a mutant, to make a mutation or to evolve this shape into another quadrangle that can be fit here. Okay, so this is something that you can either through directed means or by evolution, you can make this functionality that starts a given set of parameters, a given geometry, a given molecular structure to evolve to eventually be able to fit where you have it here. But the other trick, and this is the one that is mostly preferred by synthetic biologists, is called orthogonalization. So through orthogonalization, I remember that like orthogonal means that two systems are orthogonal when they do not influence each other. In this case, what you start it with the activity that you want to enter into this complex, and then you shape it in such a way that it will not interact with the rest of the complex, but still it will deliver and maintain its activity. So there's a kind of side aspect to this that goes as follows, and it's the problem of what is called resource allocation. So you can imagine the cell as a kind of economic system where being metabolized transactions, various things happening at the same time. And then all of a sudden, you want to enter this pre-existing and balanced system, a new component, a new actor. Then what happens? What happens is that logically there will be a competition for resources between the existing bacteria or the existing, say, molecular, say, resources that are already there and under normal circumstances are used only for cell growth, and the devices that will enter into the cells for a particular, for a specific production of a product, right? So it's like, we have a natural system, and then we want to force this natural system to follow our program, not to follow the program. And the first impact of that is that the, say, molecular and biological resources that the cells have at this point will be subject to a competition between the cell and the implants that we enter into the cell. Remember this jargon that we also developed, mentioned passing the other day. Okay, further, in many cases, what happens is that if you enter a new metabolic pathway into a cell, then some of the intermediates of the metabolic pathway are not connected to the rest of the biochemical network. And in many cases, they produce byproducts that are toxic. And then this toxicity will affect simultaneously production of biomass and also the running of the reaction itself. So you have two problems here. One of them is to deal with the competition for resources, and the second is to deal with the toxicity that is always sometimes associated to the insertion of a new pathway in a pre-existing metabolic network. So how can you handle that? Okay, so synthetic biologists are smart people, and I show you just one very, very simple example on how someone called Tom Ellis was able to handle a simple problem of handling metabolic burden and resource allocation. So look at this scheme here. So let's suppose that you want to produce a gene of interest. In this case, it's just yellow. Okay, so it's not a big deal. So you want to produce it in big amounts. But then if you produce it in big amounts, then the cells respond to it by competition, and therefore cells might be unhappy, lose their capacity to grow, and enter into a kind of bad situation. But if you enter a little circuit like this one, things can be easy. So in this case, what you have is the following. You have a T7 promoter. There's a super strong promoter that can be repressed by a regulator that is PET-R. Okay. So under no repression, this promoter goes like crazy, and then you produce a lot of PFP. But if you produce a lot of PFP, then you create a burden to the cells. how do you make sure that this gluten does not end up destroying the cells? Well, because then you have a promoter here that is sensitive to stress, and then when the cells are stressed, then they transcribe the theta repressor, and therefore this theta repressor will feed back here and will inhibit the expression of this promoter here in such a way that at the end the cells, by doing this trick, this is an artificial trick, then you can produce the amount of GFP that is sufficient for production without killing the cells because of the burden that you impose on the expression of the gene of interest. This is one of the many examples that you have in the literature on how you can handle the problem of inserting a new gene in a pre-existing network. Because cells, if you just put a gene in front of a strong promoter and activate it, the cells might end up very bad. However, if you kind of control artificially that once the cells reach a certain level of stress, then the production, the transcription is slowed down, then you can handle the situation. Okay, so orthogonality. So what orthogonal components can handle in synthetic biology? Well, you can think first, well, do nature have already produced some orthogonal components, some orthogonal parts? And orthogonal parts, remember, are those that are not influenced by the context, that they are influence each other. Well, there are some cases, not many, but there are some cases that evolution by itself has produced parts that by themselves are orthogonal. They are evolved naturally to avoid interactions with existing, say, biological systems. And one very clear case of that are the so-called the broad host range plasmids. So in nature, there are many mobile elements that can move around through different hosts, and therefore, to move between different hosts or among different hosts, they need to be orthogonal. They cannot depend on the context where they go. They have to rely entirely on their own devices. And some origins of replication of some plasmids are such that they don't depend so much on the context. They can move around. And therefore, these broad host range plasmids are excellent sources of orthogonal components for engineering biological devices. Because the promoters that are here are orthogonal, they have to work in many different hosts. The genes that are expressed, like antibiotic resistances and other genes, have to be orthogonal, they have to be active in a variety of hosts. And the replication of the plasmid has to be orthogonal in the sense that wherever it goes, it will replicate. And in the literature, in fact, this plasmid here, that is called RSF1010, is the most promiscuous plasmid known. So wherever you put this plasmid, it will replicate. If you go to a gram-negative bacterium, it will replicate. If it goes to a gram-positive bacterium, it will replicate. If it goes to an eukaryotic cell, it will also replicate there. The replication of this plasmid is completely autonomous. That's something that is good for us because it's a good source of different parts that we can get. But there are other types of orthogonal components, orthogonal parts that have an utility in synthetic biology. Some of them we mentioned the other day. For instance, if you remember, we mentioned this xenobiotic nucleic acid. And you remember that we discussed that sometimes you have alternative natural bases in DNA. You produce something that still gives the information of the DNA molecule, but it's not DNA any longer. It's something similar, but it still is functional. Or you can have polymerases from phages. Remember, we mentioned that also, that by default, in every bacterium, you have, say, housekeeping RNA polymerase, but phages, in many cases, carry with them some polymerases that are orthogonal, in the sense that they act dependently of the host. And the most famous orthogonal polymerase that is out there is the T7 polymerase, that is super strong and it's completely independent on the host. Also, we mentioned the other day these alternative genetic codes for non-natural amino acids. Remember that we discussed that you can suppress some codons and liberate or release these codons for utilization for other purposes, for instance, for loading into proteins and natural amino acids. And you're required to do that to have also orthogonal ribosomes that can detect only and translate only specific types of RNA. Remember, for instance, we mentioned that there are some RNAs that can be made based on tetranucleotides, not in triplets, and therefore the idea is that this ribosome will interpret only some RNAs and not the others. But look, we can go absolutely extreme and then think on how can we make sure that a biological system is completely independent of a pre-existing biological system. So one of the ideas that, in my opinion, are more cool or more sexy is this idea that you may find surprising about orthogonal life based on mirror components. Well, you know that every biological small molecule has a chirality. So they are amino acids are D or L, or L, you know, sugars, they have all the distribution of chiral center, DL, DL, whatever. And in biological system, that's fixed. Can we think on life forms that I would say the same structure, but are the mirror image of something that exists? For instance, what will happen instead of having something based on L amino acids, the sugars and other components, we have something that is based on D amino acids. So is there any reason why that would not work as a system? Well, this has been the kind of basis of an idea that has been developed in the last 3 years, well, already for quite a few years, and this idea of mirror life. So mirror life is this concept where you have everything that you have in the standard biological system, but you have a mirror image of everything. So if you have this enzyme, that is made with L amino acids, why not having the same enzyme made with D amino acids? Why you think that this would work? Why not, right? So if then instead of having just one enzyme, one protein, then you go little by little to all the components of the gene expression flow, and then you get mirror equivalents of each of the components. It's same. Then you may have a kind of mirror situation where this would be alive, but we have nothing to do with the other natural type of life, right? This is the basis of what has been called mirror cell. So we are not there yet, but there's considerable effort to see to what extent we could end up having a synthetic cell with all mirror components. So that means that every molecule or every protein, every component will be formed by the mirror image of the components that are in existing biological system, right? So you can have mirror amino acids, and therefore you would get mirror enzymes, or mirror DNA and mirror everything. And at the end, the question would be that you have a mirror synthetic cell. So one question for discussion, and maybe you can think about it, is whether you think that mirror an existing cell could interact with a mirror cell. Would they interact? I don't know. I give you that for your question. I don't have an answer to that. But you know that we are case of orthogonality that have this life form, I have this other life form, and they don't talk to each other because simply they are. But there are, in more practical terms, other tricks for orthogonalization of biological functions. Remember that at the end, what we have is a pre-existing biological system, and then we want to insert in that biological system a new component, a new device. So one way, a very intuitive way of handling that is to compartmentalize that new element, physically compartmentalize. Okay? And so that's very clear in the case of bacterial catalysis. So we will have a class of metabolic engineering. So this is a kind of aperitif for what will come later. But in bacterial catalysis and metabolic engineering, basically you have an input and you have an output, and something happens here. Okay. And remember that I said that if what happens here happens to be toxic for the cells because you produce intermediate toxic molecules or something that cells don't like, then one possibility is to compartmentalize, trying to isolate physically the toxic reaction or the toxic part of a pathway into a container inside the cells that will enable the reactions to happen in that container without affecting the rest of the metabolic and biochemical network. In other words, to have a microcompartment. So again, nature has already invented the microcompartments, and a typical microcompartment are the so-called carboxysomes. So carboxysomes are structures that appear inside cells, some bacteria, for instance, cyanobacteria, and they have evolved as a way to increase the concentration of enzymes involved in photosynthesis, because you know the atmospheric concentration of CO2 is very little, and the key enzyme that captures CO2 from the atmosphere is not very good. It doesn't have a high affinity, and therefore, if you just let these enzymes to be exposed to the CO2 in the atmosphere, they will not work very well. So to solve that problem, natural bacteria, cyanobacteria, have invented this trick called carboxysomes that are able to concentrate CO2, atmospheric CO2, at reasonably high concentrations to the point that now the enzymes can work very well. And this is how these carboxysomes look like. They are like small, say, containers, and inside these containers is where the action happens. So under natural conditions, these the carboxysomes contain this enzyme called rubisco. So rubisco, if you remember, or if not, I'll tell you, is the key enzyme for capture of CO2 during photosynthesis. So what happens in this case is that the structure of the carboxysome is such that it allows the entrance of CO2 in the form of bicarbonate, but when the CO2 is produced as a gas, it cannot go out. And therefore the concentration, the internal concentration of CO2, this carboxysome is very high and sufficient for rubisco to make its job. Now, the idea of synthetic biology to compartmentalize and to make this leverage this situation for the sake of orthogonalization is to engineer the shell of the carboxysomes in such a way that instead of having rubisco, you may have other reactions here. And there are various articles there and various pieces of research in which what you do is that you replace the contents of these carboxysomes with other enzymes that do interesting reactions and avoid the toxicity associated of having these reactions happening freely in the cytoplasm. But there's another approach to this physical orthogonalization of reactions, and it's the exploitation of viral capsids as nanoreactors. Well, I think that this starts giving you the flavor of everything that you can do for genetic engineering. Obviously, viruses are what they are. We all know that they are unique in some context, and so, but all of them systematically have a capsid that is formed by proteins, and in other cases, it is decorated with some derivatizations. But basically, a virus is a protein capsid. What if we separate other functions and we just keep the ability of viruses to form an empty protein shell, and then we engineer the expression of given enzyme of interest inside that shell in such a way that a given reaction of interest will happen only inside the shell and not outside. So this is something that again has been exploited, has been capitalized for the sake of synthetic biology. And then there are now a number of platforms in which you can make a fusion between the components, one protein, that is the main component, a viral capsid, and then a gene of interest, a cargo. And then when you do that and you express this plasmid in a bacterium, what happens is that inside the bacterium you form this type of thing in which the viral capsid acts as a scaffold for an enzyme or more enzymes in such a way that at the end, a reaction of interest will happen only inside the viral capsid or the X-viral capsid, and then it will not affect or will be toxic for the rest of the cell. So that's one approach to compartmentalize the whole reaction. Okay, but obviously there are other possibilities. So one other possibility is to compartmentalize the reaction, the pre-plasma. You know that gram-negative bacteria have a region between the inner membrane and the outer membrane, that's called the periplasm, where that is physically separated from the rest of the cytoplasm. So in some cases, it is also possible to export physically the reaction of interest to the periplasm in such a way that if there are toxic intermediates, this will not be exposed or mixed with the components of the cytoplasm. So this is something that is quite popular. Or finally, if you want just to avoid problems, you can just engineer bacteria that secrete the enzyme to the outside, and then you have the reaction happening extracellularly, whether completely outside the cell or whether associated to the membrane, but outside in such a way that the cell only acts as a kind of barrier for the reactions and the enzymes of interest. So this tells you that it is possible to entertain situations where the default biochemical network of the cells is not affected by the implants that we put them there, whether because we encapsulate the reactions inside with these peroxisomes or with these viral capsids, or because we export the reactions out. So these are the different tricks that synthetic biologists and metabolic engineers have to deal with this compartmentalization, with this orthogonalization of reactions. But this is not the end of the story. So the end of the story, or the beginning of the end of the story, is that in many cases we cannot export the cells, we cannot export the reaction, we cannot encapsulate the reaction of interest. I mean, we cannot do all these nice tricks that I showed you. And in some cases, what happens is that the challenge is to have a reaction of interest happening in the whole context of the cell. Okay, so for instance, in this case, you make a reaction where you have a cell and you have a reaction of interest, but the reaction of interest that might be this one, for instance, one produce molecule B, then the pathway that produces molecule B is intermingled with many other pathways, and you have constant cross-talk between the different pathways. So would be there a way to isolate in the metabolic context, okay, not with physical isolation, not with export of the reaction. Will it be possible to isolate a given metabolic module in a cell in such a way that the cell runs only these modules in a fashion that is independent of the others? So that's something that's a good question, and I'm going to tell you today a couple of different approaches to solve the problem. So the first approach is playing with conditional proteolysis of target X. And let me tell you a bit what we're. In a metabolic network, you have a network with many connections and everything, but imagine that you want to isolate one branch or one part of the whole metabolic, say, map. You want to isolate that from the rest of the metabolic reactions. So one possibility is that you identify at what points in the metabolic network this reaction of interest is connected to the others, and which are the enzymes that make the connection. So now, if you identify these reactions and then you identify the enzymes, then you can go ahead and do the following. You take the enzyme and you make a type of engineering in which you insert a protease cleavage site somewhere in the structure of the protein in such a way that in the absence of proteolysis, the cells are very happy expressing this activity, but if in a conditional form, you express a protease that cleaves the protein in a specific site and kills the protein, then that part of the connection to the metabolic network is interrupted. So this is the kind of basis, and I'll show you one example, in particular made in one laboratory, of how this may work. So imagine that you want to produce a typical metabolic map with all the connection, the curve cycle. It's just to impress people. And this is very nice, but the only reaction that I'm interested is this one. Because I'm very interested to produce this metabolite. It's called dihydroxyacetyl phosphate. It's one glycolytic intermediate, and this compound is super interesting because it's a precursor of a large number of super interesting and valuable molecules. So how can I really get rid of the rest of the metabolic network and then put together a reaction in vivo to produce large amounts of this particular compound? Okay, so this is a blow-up of the region of interest. So this is the part of the metabolic network that we want to focus on. And if you take a look to it, and maybe I will not get into the details, the dihydroxyacetone phosphate comes from splitting this 1,6-fructose phosphate into two components of the lower glycolysis. You have a 6-carbon molecule here, and then you split it, and then you form two. And there is this here, or this reaction that will make this kind of diversion of the dihydroxyacetone phosphate into this other glycolytic intermediates happens. But as I am interested to produce only this compound, the logical thing would be to make a mutation here. So if you make a mutation here, then you start with fructose or whatever, and then you will produce half of the, say, carbon that enters this compound, that then the other half will go this protein. So far, what is the problem? Is that if you make a mutant here, the cells are not viable. Okay. So it's an essential gene, and therefore, you cannot have that mutant. So how can you get some okay, this is what happens. As I said before, glucose goes into this compound there, and then the idea is that if you make a mutant, you interrupt the PIA, then cells should accumulate dihydroxyacetone phosphate. But I insist that you're going to make this mutant because you make these mutant cells die. Okay, so what can we do? Then, as I said, what you do is that you take the protein, you make some modeling, and then you try to isolate sites in the protein that are permissive and where you can insert a short peptide that is the substrate of a protease. Okay, so that's the wild type gene, a white protein under normal circumstances will work as usual. But you have these variants where you introduce in one of the sites a target site for a protease. Then, in the absence of the protease, then cells will go as normal, and then we know hydroxyacetone phosphate population. But if you have a protease, for instance, the so-called N1A protease that comes from a virus, and we'll get into that, expressed in vivo, then the protease will cleave the enzyme, and then the prediction is that you will have the hydroxyacetone phosphate population. Okay, so this is one thing that synthetic biologists face all the time. Synthetic biology is not only about making nice circuits and CFP that goes up and down. No, I mean, this is something like more complicated. So this enzyme that you see here is the enzyme called TPAA. That is this enzyme that we have to kill, okay? And the structure is known. So the question is, in this structure, how can we find regions that are permissive where we can insert a different cleavage site for a target protease? Okay, well, there are various methods. I will not go into details, but basically we found many different locations in the protein structure that tells you that if you insert a heterologous segment, a heterologous peptide, either here, then this peptide will be the substrate of a protease, and therefore, if you express that protease in vivo, chances are that the protein will be destroyed. Well, this is what happened. So this is the experiment. This is Western blot. It's an antibody that will reveal whether a protein is being produced or is not being produced. So if you take cells producing this engineered protein, in the absence of protease, and you have an antibody against that particular protein, you will see a big level of expression of the protein. But if you express the protease, then the protease you see that destroys this protein here. So we're in good shape in the sense that if we can manage conditional expression of the activity of the protein depending on whether we express the protease here or don't express. If we don't have protease, we have enzyme. If we add the protease, then it will not happen. This is all in vivo. Right? And the same happens not only with the protein, but also with the activity in vivo, right? So this is enzymatic activity. Is a measure of enzymatic activity of cells that carry this variant of TPIA that have or not been engineered with the activity site for this cognate protein. Okay? So this is what happens when you lose the activity. So you have here, and then this is what happens when you have this conditional enzyme in the presence or absence of the proteins. Okay, so in the absence of the protein, you have a high activity here. In the presence of the proteins, you lose the activity. So I mean, this is just an illustration that can really make this type of conditional expression of different enzymes by doing this. Now, what happens then is that in the absence of in the normal wild type strain, you have no accumulation still phosphates. In the mutation that you get there, iron more, and when you have the protein that is engineered to be conditionally dependent on the protease, you see that with the protease, indeed, you have more differentiation. Okay, so that's in principle the basis of this portabilization. And now the next problem is how to bring about a very, say, tight control in the expression of the protease. This is another level of problem. So I'm saying with you today just a very practical case of how to engineer an orthogonal system, not only conceptually, but also for the point of view of trying to implement this in real terms. Okay, so when we face the challenge of making a genetic construct, then we have to consider what expression system we want to use. There are many expression systems. The most popular are based on IPTG. This is the most famous one because IPTG will induce promoters that are controlled by the lac repressor. But there are many others. And one of them that is very good is one that is run by this transcription factor that is called SIDE-S that activates its cognate promoter in response to 3-methylation. Well, in fact, if you what you want to have, a switch between full activity of the enzyme and no activity of the enzyme, is to have a very strong switch. Either you are on or you are off. You don't capture intermediate levels. So to kind of handle that, you have to be very careful with the basal levels of expression that you have in the absence of expression. And then in the absence of induction. So we have here, for instance, a list of 5 different expression systems that you can find in the literature based on LAC-I, based on SILES, PPRK. I mean, if you look at this, it's like a shopping list. So you have a whole repertoire of possibilities for bringing heterologous expression of a given gene, and then you make your choice. And then out of them, if you look at the basal level of all of them, then you notice that by looking at this, well, this has quite basal level. I don't want that. I don't want this. It has auto basal level. I'm not interested. This is nice. It has very low basal level, but it's the user is very weird because it's an aromatic compound. It's a chlorophenol. I'm not interested. The other is activated. Is well controlled, but it's activated by a volatile inducer. Very weird. Okay, so let's focus on this one that has a reasonable level of, but sample basal level, and we see what happens. Okay, so the gene expression model that we use for that is this one. So remember yesterday or the other day, the basic model for genetic devices in synthetic biology involves first this biosensor model. In fact, this is not very bad. This is a way of measuring this experiment with a technology called cytometry, in which you measure what is the expression level of each cell in a population. So, and this is a population of cells that have been engineered with this device here together with DNP. So you see that in the absence of induction, cells have very little, say, expression levels. And as you induce the system, then the population starts moving in this direction. And under full induction, there's no radial overlap with no induction here. So in principle, this system looks quite good to this. Okay, so let's make a construct and see what happens. So what happens is this. So you make a plasmid, and in one plasmid you have the proteins under the control of the system, side S and BM, and what is the plasmid. And then in the other plasmid, you have the gene of interest, in this case TPA, that has been engineered to be cleavable by the protease. Okay. Now it comes the experiment. So this is the control with the cells that carry plasmid with the enzyme here that cannot be cleaved, so it remains there. And look at this. So when you have a cell that carries these two plasmids, then in the presence of the inducer, then the protein is destroyed because that is cleaved. But even in the absence of inducer, then there's a considerable amount of enzyme that is cleaved. So this is really bad because we want to have a switch, and this is not a switch. No, we have the control. We want to go directly from here to here, but we don't want to have this intermediate step because it will build the type of switch that we are after. So how can we handle that? So you start with something that is not digital. How can you bring about digital expression? So in other words, most promoters have this pattern here. Most heterologous expression system. So you start with a given level, but the level is not zero. And then you have induction. But what I want is zero, because if I don't have zero, then forget it. Then how can we bring this down here?